Welcome to Lecto. Today we are going to discuss passive transport. What is passive transport? Passive transport is that transport in which the molecules move from higher concentration to the lower concentration without any usage of energy. So it has different types or categories. So first of all we will discuss the simple diffusion. Simple diffusion in which the molecules move from higher concentration to the lower concentration without any use of energy. So, simple diffusion can occur through either by lipids or through protein. So, first of all, we will discuss the simple diffusion by the lipid. As we know, here is a cell and having nucleus in it. So this cell is surrounded by the plasma membrane, or you can say cell membrane. Cell membrane has two basic components, proteins and the lipids. So here you can see this is the structure of the cell membrane, and this is the layer of lipids, and this is bipolar lipids. And here you can see there are different substances present outside the membrane, and they are in high concentration. These molecules move through the lipid layer to the low concentration, from higher concentration to the lower concentration through lipid layer. Those molecules can cross the lipid layer which are soluble in the lipids like carbon dioxide, etc. So these molecules can move through the higher concentration to the lower concentration, and this is called the simple diffusion through the lipid. Next, simple diffusion through the protein. Simple diffusion through protein. Again, molecule move from higher concentration to the lower concentration, but there is an involvement of protein. It has further two categories, ungated and gated. Ungated proteins are actually embedded in the plasma membrane. They are actually transmembrane protein. Those proteins which are actually embedded in the lipid layer, lipid bilayer. Here you can see this is the lipid layer, and this is the protein. So this protein has no gate, and it is freely and molecules can freely move through this lipid. Those molecules which cannot cross the lipid bilayer can adopt this path, and those molecules will move through these protein channel from higher concentration to the lower concentration. So these proteins has no gate. But if we talk about the gated protein, it has further three categories: voltage gated, ligand gated, and mechanical gated. Here you can see, first of all we will talk about the voltage gated. Here you can see, if we talk about the example of sodium channel. Sodium channel. So these proteins, or you can say channels, has three forms, or can exist in three forms. First of all, if we talk about the sodium channel, then this channel has two arms. Here you can see this is the inactivation or inactivation gate, and here you can see this is the activation gate. So when the activation gate is covered the channel, then it will block temporarily, and it close, and the molecules which are present outside will not pass through this channel. This is the first form. Second one is if both the gate are open, then molecules which are present outside in higher concentration can easily move into the cell. But if the inactivated gate is cover the channel, then it will permanently block. It will permanently block, and the molecules which are present outside the gate will not pass into the cell membrane. So these are basically the three forms. First one, if the activated gate covered the channel, then the molecules cannot move into the cell for temporary. But if both the gates are open, then molecules can easily pass through the channel. But if the inactivation gate is covered the channel, then the movement of the molecule will permanently block. So next we will discuss the ligand-gated. What is ligand? Ligand is actually a molecule or a substance which actually bind the protein channels. Here you can see this is the ligand-gated channels or proteins. Here if we talk about the neuron, here is the neuron and this is the axon. This is the cell body and this is the axon. Here another neuron which is present, and the two neurons are joined by the synaptic clefts. Neurotransmitter which release from the axon and move into the synaptic cleft will enter into the dendrites of the next neuron through the ligand-gated channel. Here let's suppose you can see there is a neurotransmitter, or you can say acetylcholine. This is the neurotransmitter. When this acetylcholine will bind to the postsynaptic membrane of the next neuron, it will bind this ligand-gated channel. And when this acetylcholine bind to these proteins, then the gate of these proteins will open and the molecules which are present outside, let's suppose sodium, will start moving inward. So these are basically the ligand-gated channel. These channels are open only when a ligand bind to these channel, and after binding the gates which are present over the proteins will open and the molecule start moving inward. Next is the mechanical gated. These gates are open only when there is a stress. So mechanical gated channels open in the presence of stress. So let's suppose if we talk about the Pacinian corpuscle. These are basically the corpuscles or the receptors which detect the pressure in the skin. And when here is the channel, mechanical gated channel, and there is a gate present over this channel. and when the pressure applied on the skin, then this gate will open and the molecules which are present outside, let's suppose sodium, will start moving inward. So these are basically the mechanical gated channels which open only when there is stress. If we apply stress, then these gates will open. So these are basically the three types of the gated: voltage gated, ligand gated, and mechanical gated. Next, we will talk about the facilitated diffusion, or you can say carrier mediated diffusion. There is a difference between the simple diffusion through protein. These are protein, but these through facilitated diffusion, these are also protein, but there is a difference. If these proteins, gated proteins, have channel, and facilitated diffusion or carrier mediated diffusion, in these carrier proteins has no channel, then why it's mean ungated and carrier facilitated diffusion or carrier protein is same? No, in these protein there is no transformation in the structure, but in facilitated diffusion there is a change in structure, and through its changing its structure, the molecule can easily move across the plasma membrane. Here you can see these are basically the carrier protein. These are basically the carrier protein, but these proteins are channel. These are channel protein and these are carrier protein. Let's suppose the glucose molecule which are present outside the membrane in higher concentration. These are basically the glucose molecules. Glucose molecule will attach to the carrier protein. When it will attach to the carrier protein, then there first of all it will attach. Then there is a change in conformation of the protein. So here it start changing its shape, and after that the molecules of the glucose will move inside the cell. So this is actually the facilitated diffusion which help in the transport of the molecule, but in that situation again there is no use of energy. So this is actually the facilitated diffusion. And the fourth one is special time. It has further three categories: bulk flow, filtration, and osmosis. In bulk flow, as name indicates, there is a huge amount of substance will move across the membrane. We will discuss the example of alveoli. Here is the lungs. This is the bronchi, bronchioles. And in lungs, there is a alveoli. Here you can see this is the structure of the alveoli. Alveoli. And lot of blood capillaries surround these alveoli. As we know, the air which moves into the alveoli has higher partial pressure of oxygen, and the oxygen will start moving into the blood from high partial pressure of oxygen in the alveoli to the low partial pressure in the blood capillaries. And in the blood capillaries there is a higher pressure of carbon dioxide, and in the alveoli there is a low pressure of carbon dioxide. So carbon dioxide will move from higher pressure to the lower pressure. So bulk flow is actually depend on the difference in the pressure. Oxygen pressure is higher in alveoli, and in blood capillaries there is a low oxygen pressure. So oxygen will start moving from higher pressure to the lower pressure. And here again this is reverse. Carbon dioxide has high pressure in blood capillaries and low pressure in the alveoli. Then the carbon dioxide will start moving from blood capillaries into the alveoli. So this is the exchange of the carbon dioxide and the oxygen. That's why we take oxygen inside our body and exit or exhale carbon dioxide outside the body. So next, this is actually the bulk flow. Next, if we talk about the filtration, filtered something. This is the nephron, and here you can see this is the glomeruli. This is the structure of glomeruli, actually the clusters of different capillaries, and here it has different portion. This is the Bowman's capsule, this is the proximal convoluted tubule, this is the loop of Henle, this is the distal convoluted tubule, and last is the collecting duct. So this is actually the structure of the nephron. If we talk about the glomerulus, this is the afferent arterioles. Afferent arterioles enter into the glomerulus, and afferent and efferent arterioles. Afferent arteriole enter the glomeruli, and efferent arterioles exit the glomerulus. When the blood moving towards the glomerulus, and there is a pressure which exert on the glomerulus, and the substances like water and different minerals start moving into the Bowman's capsule. But keep remember, proteins and the bloods will remain in the glomerulus, which will remain in the blood. But the other substances like water and different electrolytes will start moving into the Bowman's capsule. So this is actually the filtration in which the substances will move from higher concentration to the lower concentration. So next, if we talk about the osmosis and other special types of the passive transport, in which the molecules of the water move from low solute concentration to the higher solute concentration. Here you can see these are basically the four tubes: tube A, tube B, and tube C. Tube 1, all these tubes having semi-permeable membrane in its center. So tube A has water molecules, and their level are equal. So tube A has only water in it. But if we start adding some solute molecules or particles, let's suppose sugar, in the right side of the tube, then what will happen? Now the right side of the tube has high solute concentration relative to the left side. So water will start moving from the low solute concentration to the higher solute concentration. So this is actually the osmosis. Water molecule will start moving from low solute concentration to the higher solute concentration, and it will start moving upward, and it exerts a force in upward direction. Let's suppose 20 Newton force will exert in upward direction, and here the water start moving down, and here the solution start moving upward. So, and the difference which create between them is called the osmotic pressure, and this difference is called the osmotic pressure, which exert in the upward direction. So if we put a piston over there and prevent its movement in upward direction, if we put a piston and start or prevent its movement in upward direction, and then it will stop the osmosis movement of the water molecule from the left side to the right side, and this another terms, or which is also called the osmotic pressure. Here you can see here this is a person, and this person drag the box with a 20 Newton force and it will displace in the forward direction. But if another person exert a force of 20 Newton, then this box will not move. It means if we apply equal force 20 Newton force on the box, then this box will remain static. So here is the thing that 20 Newton force apply in upward direction. If we put another 20 Newton force, same force, then due to the same force, the osmosis or the movement of the water molecule will stop, and this is called the osmotic pressure. And we calculate osmotic pressure on the basis of the applied force, because both forces are equal. Either it will start exerting in upward direction or in the downward direction, so it's easier for us to calculate the applied force. And here another terms which is called the reverse osmosis. Reverse osmosis. So another important osmotic pressure, if we talk about, is osmotic pressure. If the large molecule start exerting pressure, then it is called the colloid osmotic pressure, or you can say oncotic pressure. Like blood has lot of albumin, which is large molecule, and due to these solute particles, the force or pressure exerted by these solute particle like albumin is called the colloid osmotic pressure or oncotic pressure. So here is the different molecules, water molecules present in it, and here is the solute molecules, and there is 20 Newton and we apply 20 Newton force, and now the situation is in equilibrium. If we exert 10 Newton more force on the piston, then a state reached when the water molecule which are present on the right side will start moving towards the left side, and this is against the osmosis. This is the movement of molecule from higher concentration to the lower concentration. This is actually the reverse of the case B, in which the solute molecule move from low solute concentration to the higher solute concentration, but in that situation the water molecule will start moving from higher concentration to the lower concentration. This is called the reverse osmosis. So this is all about the passive transport. In this lecture we discussed 4 different categories: simple diffusion through lipids, simple diffusion through proteins. It has 2 categories, gated and ungated, and gated has further 3 categories: voltage gated, ligand gated, and mechanical gated. And next, facilitated diffusion, and last one is the special types. It has further 3 categories: filtration, bulk flow, and the osmosis. So this is all about the passive transport. If you have any question, then you may ask in the comment section. Thank you.